Good day. My name is Dr. Jennifer Giandhari, and I am a researcher at the KwaZulu-Natal Research and Innovation Sequencing Platform, which is based at the University of KwaZulu-Natal. I'll be presenting on a study which looked at the frequency of mutations in the 3' polypurine tract of NEF in people with viremia on dolutegravir-based art across HIV subtypes in Africa. This study forms part of the IDEA DTG Resist project. Mutations within the integrase gene, which have been associated with dolutegravir resistance, have been well characterized. However, there have been some studies which have reported on mutations occurring outside of the integrase gene. These have been associated with resistance to DTG. These studies have found that mutations in the 3' polypurine tract of the NEF gene in viremic patients may affect DTG effectiveness. However, at this point, the frequency and significance of these mutations in NEF remains unknown. We do know that the 3' polypurine tract region in NEF is well conserved, so we extended our analysis in the study to also include the 3' and 5' flanking regions. The study examined the frequency and distribution of these mutations across various HIV-1 subtypes in Africa. We performed viral load testing on plasma or dried blood specimens. We then performed HIV whole genome sequencing on a subset of specimens with a viral load greater than 5,000 copies per mole using probe-based hybridization with the X-Gen HIV-NGS hybridization capture panel from IDT. We then aligned our sequences to the HIV reference strain HXB2 and identified mutations in the 3' polypurine tract region of NEF, as well as in the flanking 3' and 5' regions. The mutation frequency was assessed overall and by HIV subtype. Just to orientate you, the slide shows the subtype distribution across the sequences that we looked at in this subset. We had a total of 196 sequences. It is important to note that of these, majority were subtype C, with a smaller number of sequences among the other subtypes. The sequence shown here refers to the 3' polypurine tract. These are the nucleotides which are located at the bottom of the slide. So the bottom of the slide is relative to the NEF region, and then you have the flanking regions on either side. The nucleotide positions have been placed under each subtype diagram as well and correspond to the diagram below, which is the region that we studied. For this figure, if you focus on just the 3' polypurine tract region, which is the 4 consecutive A's in green, ending with the 6 consecutive G's in orange, you will see that it's relatively conserved throughout the different subtypes, except for subtype D. However, this mutation occurred in one of the 3 sequences. We then expanded our analysis to the flanking regions, where we can see some degree of variability, especially if you look at the second nucleotide, which is a CT for subtype A, as well as the CRF and the recombinants. This table focuses on 5 sequences in particular, which had changes in the 3' polypurine tract region. All 5 of these came from treatment-experienced patients who were on TLD. Of these 5, only one sequence had major INSTI mutations as well as NRTI drug resistance mutations. From the study, we found that mutations within the 3' polypurine tract region of NEF were rare, with only a single case detected in subtype D and C. and 3 in recombinant samples. However, mutations in the regions flanking the 3' polypurine tract were more frequent and appeared to be associated with HIV subtype. These findings suggest that the observed variability is likely due to natural diversity. Further genotypic and phenotypic analysis, as well as clinical correlation, is needed to understand the significance of these mutations and to clarify whether they contribute to virological failure in the absence of integrase drug resistance mutations. Thank you.